Hello folks, let's summarize the process of protein synthesis. Protein synthesis can be described or summarized in terms of what is called the central dogma. The central dogma is, in essence, DNA to RNA and RNA to protein. That's the central dogma. It includes the processes of DNA coding for proteins. We know that protein synthesis occurs on ribosomes, which are outside of the nucleus. And remember that DNA is held in the nucleus. DNA has to make mRNA to transfer the information, the genetic code, to the ribosome. And the central dogma summarizes these steps. So, here is this DNA to RNA and RNA to protein in an illustration. Remember that the DNA molecule is where the gene is located. And a gene is a segment of DNA. It's a piece of DNA that codes for a protein. And you know that proteins are amino acid chains. So we have a DNA template, in other words, a DNA pattern. A DNA pattern. A, C, C, A, C, C, G, A, G, T. These are DNA nucleotides. These letters are the nitrogen bases that are on the nucleotide. We start off with a DNA template, a DNA pattern. That DNA is then transcribed into mRNA. The process of transcription. Transcription takes place in the nucleus. The product of transcription is mRNA. Notice how the mRNA molecule has nucleotides as well. It has nitrogen bases. All right? And they are complementary to the DNA. So whatever the DNA is, the mRNA will be the complement, or the one that matches. And please remember, it's very important, that if your DNA strand has adenine, the complementary mRNA will be uracil, U. So if you have A as your DNA, you're going to have U in your mRNA. And then everything else is the same. C pairs with G. Again, A pairs with U. There is no thymine in mRNA. There's no thymine in RNA, period. C pairs with G. G pairs with C. Again, A pairs with U. G pairs with C. And thymine, if thymine's on DNA, that pairs with adenine. There is adenine on mRNA. Okay, that's transcription. We talked about that during class. We also talked about translation. Once you have your mRNA molecule, every 3 letters on the mRNA is called a codon, and each codon brings in, or is specific to, an amino acid. So every 3 letters on the mRNA gives you an amino acid. And the sequence, or the order of codons, brings about the order of amino acids. And the order of amino acids determines the function of the protein. Remember that amino acids are the monomers of proteins. Amino acids build proteins. And the order of amino acids determines what the protein will do. So let's summarize transcription. We're going to start off with that. Summary of transcription. Transcription occurs in the nucleus of eukaryotic cells. Transcription makes mRNA from a DNA template. It uses complementary base pairing, but remember that RNA does not have thymine. So if your DNA sequence is CGTACTAGCGAT, sorry about that. If this is your DNA sequence, Your mRNA, your complementary mRNA sequence would be what pairs with C, G. What pairs with G, C. Remember, this is mRNA. What pairs with T, A. What pairs with A, U. There is no thymine in mRNA. If you are making an mRNA molecule, if you are making an mRNA sequence from DNA, you do the complementary base pairing, but remember that adenine pairs with uracil. So let's look at this. Take out a piece of paper. I want you to write down these 1, 2, 3, 4 sequences. Write them down. Give yourself a little bit of space because I want you to write the complementary mRNA sequences for each one. Go ahead and pause the video, and I want you to try that. Okay? So what you're going to do is you're going to look at the DNA sequence, and then underneath it, you're going to write the complementary mRNA sequence. Let's look at the first one. What pairs with C on mRNA? G. What pairs with T on mRNA? T would pair with A. What pairs with A? U. We're doing mRNA. We're making an mRNA strand. Please take some time and do all 4 of these. 1, 2, 3, 4. Go ahead and pause it. Here are the answers. Let's check your answers to these 4. Your mRNA sequences should be this. Check it out. If you have any questions about this, shoot me an email and let me know. Okay? So again, let's look at transcription again. We looked at this during class. Here is your DNA. This is your DNA molecule. Remember that transcription, is done using an enzyme called RNA polymerase. RNA polymerase attaches to the DNA molecule, and it attaches complementary RNA nucleotides to the DNA strand, using DNA as a template, DNA as a guide, DNA as a pattern to make the mRNA. And what do you end up with at the end of transcription? You end up with mRNA. Now, the purpose, let's look at this video. The purpose of transcription is to take the DNA and make an mRNA copy. Okay? So you're taking the DNA molecule, this is an animation of DNA, and you're going to have an enzyme do all these things. So terminator region, coding region, you don't have to worry about. Promoter site, that's where it all begins. That blue blob is RNA polymerase. RNA polymerase will separate the DNA and attach RNA nucleotides to corresponding DNA nucleotides. It's going to use the DNA as a template. The DNA is used as a pattern. So that blue blob is making what? What's it making right now? It's making mRNA. That mRNA is then going to go to the ribosome in the cytoplasm. DNA codes for proteins, but protein synthesis happens in ribosomes outside of the nucleus. DNA can't leave the nucleus, and so you have to make mRNA to move the information from the DNA to the ribosome. The information for making proteins. And that's transcription. DNA is the template, the pattern, the guide for making mRNA, messenger RNA. And the messenger RNA goes into the cytoplasm, where it then meets up with the ribosome, and then the next step of protein synthesis takes place, which is translation.